Here we go. Hey, Sid. Okay. So we'll be talking about structure and function today. So kind of following up on our Professor Dave stuff. So Sydney, and I don't know if Savannah's with you. I think she is. Who else is here? Is he here? Okay, yeah. So after setting up a lab, which was really fast today, we spent some time watching some really good Professor Dave videos. I'm going to link them in the email. You guys notice that in the weekly email at the bottom, there's like every video that either we watch in class or that there's some extra good ones there. They're really good, especially when you're studying. So next week is our last plant lesson. We'll be wrapping up plants. So then we'll have a test, and it's always, those are always good to watch because they'll help remind you of what you're doing, what you're studying. I'm going to grab myself a cheese stick and then we'll do this.
No, I think that's
I'm
the door. Right.
okay. Hi, Boo. I like it.
I like imagine that's like I'm like, I don't know what this
So y'all probably noticed before lab that Jason has hooked up a Bluetooth speaker into the giant phonograph horn in there. That's what that was. The wires are not hidden yet, but we're working on it. So I was joking last night. I said I should play Reveille to tell all my kids it's time for lab. And Drew was like, yes, do that, please. Just, it sounds great when you play like old school music on it. Like Jason was playing the Andrews Sisters and stuff like that. So it was doing Boogie Woogie Bugle Boy. But modern music sounds weird on it because it's
Jive and Whale.
Jump Jive and Whale would be good. Yeah. Well, Brian
put
Setzer.
that on the speaker beneath it,
Mm-hmm.
but then I realized it was
Coming from the horn.
I was like, what?
Yeah, he hooked up the Spy, the Amazon Spy to it. So she responds from in there too now. So if you ask her any question in there, she answers through the horn instead of through the speaker. Yeah, you know, we're weird like that. All right. So lesson 21. We are again going from microorganisms to plants. We're about to wrap up plants next week. And then we'll be on to animals. And then after animals, we're on to humans and we're done. So let me see. Do I have anything here to hand out? I have something for Ethan. I have something for Shannon. And something for the Ames aren't here. Yeah, I have one thing for you. This is Ethan. What's your stuff? That's for you. All right. All righty. Oh, and Jack. You're just hiding underneath it. So plant structure and function. That's a cross section of a stem, and it's just really pretty, isn't it? I like that. Looks like a stained glass or something, right? Okay, so homework, lesson 20. Here's our review for homework. Most plants are flowering plants, true or false?
I can't
True.
Yeah, remember what you wrote? It's true. 80% of plants are flowering plants. Okay, what is the second largest group of plants?
remember. Second largest group.
Green algae.
Do you remember what you put? Yep, green algae. Very good. All right. And remember, this is the one, green algae is the one where some green algae ride that line between protists and plants, right? That's kind of, when you see things like that, that's your link, your evolutionary link really between one group of organisms and another. You hear about that, where's the link? That's the link. All right, how about, why are bryophytes small?
Because they don't have roots.
They don't have roots. What else don't they have? Vascular tissue. They don't have roots and vascular tissue. Why does that make them small?
Because then they don't have any way of getting nutrients long distance.
Yep, they can't move nutrients and water far, so they can't get so big. That's exactly right. All right, now how about what do you see in this picture? There was the little picture. Do you remember it? What do you think happened? It was this picture. Do you remember?
Looked like pieces of pine cone. I said that
Did.
squirrel ate a pine cone.
There you go. It did look like pieces of pine cone. So that is what happened. So squirrels will eat both the nuts and the outside scales of a cone, making it a whole meal. And while unripe green pine cones have the delicious seeds and the tasty outside scales, the squirrels will still eat mature brown cones if they're the only ones they can find. Green pine cones have high nutritional value and are a great source of fiber and different vitamins for squirrels. They play an important role in their diets That's why squirrels like to store green pine cones to ensure that they always have these nutritional snacks close by. You ever seen them run off with them in their mouths? Yeah, they'll run off with them in their mouths and tuck them into their little nests wherever they live. Hang on to them for a while. They'll bury them too, I've seen. They'll bury nuts and they'll bury pine cones. So this picture here. It was a really beautiful picture of these flowers, right? And it said, I want my awesome flower garden to grow back next year. Did I choose the right kind of flower? Did anybody get this one?
I don't.
I did.
these
I
questions.
answered.
Don't remember what you wrote down?
I think I said. I had my mom.
I
come
think
in and
I
she was
said
like, "Those
no."
are petunias. Those aren't gonna grow back."
Those are petunias, right? Okay, so whoop,
Yeah, I
Hello.
asked my mom what type of.
what they
flowers
are.
were.
Good. So those are petunias.
Technology.
Petunias are annuals. They will not come back next year. So that somebody put a lot of effort into that beautiful display, and it's going to be 1 year.
I also researched the picture.
Mhm.
Got like Dubai gardens or something.
There you go. So this common house plant is called a sago palm, but it's not a palm at all. Why isn't it a palm? And to what division of gymnosperma does it belong? What does it have in common with the palm? Anybody know what it was? It was a type of gymnosperm, but it's not a fern. It's a cycad. Remember cycads?
No.
So what do cycads have in common with palms? Well, their leaves are similar.
They're
And they-
both gymnosperms.
And they have that, well, no, palms aren't. But when a palm tree grows and the leaves fall off, it leaves a scar on the stem. And the same thing happens with cycads. So the answer is sago palms, which isn't a palm at all, is a cycad. So they're an ancient division of gymnosperms. True palms are angiosperms. Both gymnosperms and angiosperms produce seeds, so they have that in common. All right, and their seeds, sago palm seeds, are in that cone, just like they are in conifers. So specialized tissues. So now we get really into the nitty gritty here of how these specialized tissues work. Now, just so you guys know, in the next couple of weeks, when we move into animals, your lessons are going to be kind of weird. They're going to be different than they have been. I don't know if you flipped ahead and looked. But the way the juniors was set up is it went through types of organisms from the least complex to the most complex. So it starts with, invertebrate, non-chordate organisms, then it moves into chordates and then vertebrates, and then that breaks it down into, your fish and amphibians, your reptiles, your mammals, that kind of thing, birds. Your book went more into systems, animal systems. So what's going to happen with your lessons is you're going to see your lesson from your book, which is going to be all about animal systems. It may be reproduction and respiration and excretion in your lesson, but then it'll also say mollusks and arthropods or whatever. So that will be in that week, you will read about your systems, and then you will you'll have a little thing at the end of your lesson in your binder. That's the same thing the juniors are reading, and it will cover that animal type. So it'll be like, this is on mollusks, this is on arthropods, and that's not in your book, but you're going to need to read that. Okay, so I brought that up because the specialized tissue thing reminded me. So anyway, so I was like, oh yes, your book gets very specialized. So that'll be important for you to read because your labs are going to align with what kind of animal it is, because that's when we get into all of our dissection labs. So specialized tissues. All right, back to plants. We have 3 tissue systems in a plant. You have dermal tissue. What does dermal mean?
Skin.
Yeah, skin. If you see derm, like a dermatologist, whatever, derm has to do with skin. So dermal tissue, vascular tissue, we know what that is, and then ground tissue. What's ground tissue? Do you know? Do you remember?
I have an idea that is almost certainly not correct.
Okay, what?
It's tissue that's used to ground out electricity.
Yeah, there you go. Ground tissue is basically the stuff in between. It's the stuff in between your dermal tissue, your vascular tissue. It's kind of like the base, or whatever you want to call that things are building on. And then you have 3 organ parts of a plant. Do we know what they are? Professor Dave talked about it. You got roots, stems, yeah, and then leaves. Now remember, flowers are modified leaves. So these are our 3 major organs in a plant. Plants have organs just like we do, and these have systems within them. So a special type of tissue called meristematic tissue is the only plant tissue that can produce new cells by mitosis. When you see this stem in here, it's stem cells, just like when we had back at our platyhelminths, our flatworms, right, our planarians. We cut them in half in certain places where they had stem cells and they regenerated body parts that were specific and differentiated. And remember, we talked about how there are stem cells in bone marrow, right? Because it can differentiate into different kinds of cells. And then you have stem cells in your body in certain places where you can grow specific types. It can differentiate into specific types of cells, right? So stem cells are very important because without them, you just kind of keep producing clone cells, right? So meristem cells are going to be in specific places on a plant. This is why, with some plants, I don't know if you've ever done any like trimming of plants in your yard, but sometimes like your mom might say, hey, if you're going to cut that rose, don't cut it until you see the 5-leaf branch instead of the 3-leaf branch. And that has to do with where the meristem cells are located. And it'll cause the plant to get bushier or get taller or whatever. So if they're found in the tips of the shoots and the roots, and they're also found in some places along the stems and leaves, depending on where. So our dermal tissue is first here. So this is a good graphic. It's a really, really good graphic. And you can see it's broken down. You've got your cuticle and your upper epidermis. Last week, we tried to remove upper epidermis, somewhat successful. Not at all with that water plant. That was really hard. And then you can also see your lower epidermis, which we also tried to remove. So these are the outer coverings of your plant. And then there's this cuticle. Remember, the cuticle is just like that waxy material that keeps the moisture in. And then there were these guys. This is what we were looking for last week, these stoma. Okay, these are little holes, right? Little tiny holes in the leaf. And these guard cells control how open or closed the hole is. And they were really easy for us to find last week on the lower epidermis of our land plant, right? Like we opened up that cherry laurel and that lower, spun that microscope head around and boom, there they were. It was really, really obvious where they were. It was pretty cool. And then you also have all kinds of different cells in here, and they're broken down into palisade cells and there's spongy area in here, and they call this whole thing a mesophyll. So you got an outer covering of your plant here, and then it consists of a single layer of epidermal cells. So you see how these cells are kind of squashed? This is a cell. Each one of these is a cell. So this is your outer layer in your upper epidermis cells, whereas these cells are much different, right? These cells are like vertical, and they call them palisade cells because it looks like a do you know what a palisade is? It's like a how do I describe it? Like a walkway, usually with like a very vertical looking. So that's why those are called that. Then you got your cuticle, your waxy layer, right? And then you've got the purpose of your cuticle is to provide against water loss mostly. And I don't know why that dot's by itself. Okay. And it may include specialized structures such as root hairs and guard cells. And here's your guard cells. And those were super important. So then you got your vascular tissue. And we talked about this a little bit last week. You got xylem and phloem. Does anybody remember the difference? What's xylem do?
What moves water doesn't move nutrients.
Right. Moves water primarily from the roots to the shoots and leaves, right? It's the one that goes up, right? And then what's the phloem do?
Yeah, moves nutrients down.
Not always down, but yeah, it's what's taking the nutrients, specifically the sucrose a lot of the time from the photosynthetic part, so from your leaf, right? And then moving it around the plant. Sometimes it's down, sometimes that's somewhere else, but it's helping the nutrients flow from one place to another, right? And what's it called when you move nutrients from one place to another? Do you remember?
Nutrient transport.
Translocation. So that's one to remember, translocation. So it transports water and nutrients throughout the plant. Your xylem transports water, and it's composed of dead cells. Like it's kind of like when you think about fingernails or hair, you've got like this dead fibrous thing. That's kind of the core. That's the pipe that they've made here, is this dead cells thing. So your phloem is going to transport your nutrients, composed of living cells, and they're called sieve tube elements. So basically, you know what a sieve is, right? Like a colander, you can strain through it.
Yeah.
That's the idea with phloem. It allows things to come in and out through these little structures called sieve tube elements. So your ground tissue, this is what we're talking about a minute ago. Your ground tissue is your stuff in the middle, really. It's the stuff in the middle. So it lies between the dermal and vascular tissues. So we have parenchyma, right? Here's a great example. It has thin cell walls and large central vacuoles. What would you think something with a large central vacuole would Like what kind of a structure? Do you think it would be a floppy, squishy thing? Do you think it would be more rigid thing? What do you think? Vacuole is going to hold the water, so yeah, it would definitely be more rigid because that vacuole is like a water balloon in there, right? So it's going to give it some rigidity. It's going to give some turgor pressure. We talked about turgor pressure. So here's parenchyma. There's an example. You can find parenchyma in potatoes. So it's got this intracellular air space. So you can see where these cells don't touch, and there's actually air in there. And then this kind of shows sort of a longitudinal. So this would be, I guess this one is cross. Yeah, this is cross section and this one's longitudinal. So if you took a piece of potato, you cut it one way and you look down through the top, then you flip it and you look up at the side, this is what you're going to see. All right. Next one is the collenchyma. And here's your example is your celery. You've seen celery many times. I'm sure you've looked at it. Cross section of it, you've got these nice little vertical things. Thick primary cell walls, right? And it's strong, it's flexible, it's found in larger plants. And then sclerenchyma. This word here, scleren, you'll see this other times too. It means like tough. Scleroderma is a disease some people get, causes their skin and other exterior tissues to get really, really rigid and hard. And actually scleroderma is a terrible disease, but so sclerenchyma, extremely thick, rigid cell walls. So this is where you get really strong structures, right? So when you think of sclera or ever see that, it always has to do with rigid or hard or firm. So for your work, it said list 3 principal organs of seed plants. So they were what? Roots, stems or shoots, yeah, and leaves, right? Roots, stems, or shoots and leaves, right? Always. So what adaptation helps leaves conserve water? You can think of 2 right away. The stoma is going to control how much water goes in and out along with gases, and the cuticle, that's going to be your big one. So your 3 main tissue system of plants are going to be your dermal tissue, your vascular tissue, and your ground tissue. So your dermal tissue is going to be where in the plant? On the what part? Yeah, on the outside, right? Your vascular tissue is going to be on the inside, and then where's your ground tissue?
Everywhere else.
Everywhere else. Exactly. Very good. It's just kind of surrounding everything else. All right. So the cuticle protects against water loss, which we just talked about. Some epidermal cells have tiny projections known as trichomes, which give a fuzzy appearance. So when you see leaf that's fuzzy, that's called a trichome. Dermal tissue in roots contains root hair cells that help absorb water. So if you look at a root really close up, you'll see it has these little hairs on it. Yeah, it's cool. It's really cool. So let's see. Trichids are long, narrow xylem cells. Lignin is the substance in cell walls of dead trichids that make wood tough. Let's see what's next. Xylem cells arranged end to end on top of one another are vessel elements. This is just vocabulary that you guys did. And here's your sieve tube elements that we talked about. Those are your main phloem cells. And then your companion cells are cells that support your phloem and acid in the movement, in the mood of substances. Wow. So how can water move from one trachid to a neighboring cell? Cell walls of tracheids have openings that connect neighboring cells one to another. And then how can materials move from one sieve tube to the next? The end walls of sieve tube elements have many small holes, and that's why it's called a sieve, right? Right. So your parenchyma, we looked at that a minute ago, and your collenchyma we looked at, and your sclerenchyma. So meristems are regions of the plant that produce new cells by mitosis. Is that true or false? True. That is true. It has the word stem in it. It's what kind of cell?
Stem cell.
What does that mean? Stem cells can do what?
Differentiate
Into
other cells.
any kind of cells, right? Right.
Is it innate or?
Typically, but not always. I mean, obviously it's not going to become a bone cell in a plant, but whatever those stem cells are capable of becoming, which is more than one. Because if it's mitosis in a leaf cell and it just makes another leaf cell, right? Because that's how mitosis works. As those aren't stem cells, but when you have stem cells, it can say, well, I'm going to make a leaf here. I'm going to make a stem there, right? So apical meristems are found in the growing tip of a root or stem, and that's true. Apical or apical, what's an apex of something? Like
magnum opus.
Okay, explain.
Yeah, I mean, like the grandest part or the most important or the
Mhm.
most,
The top.
Yeah, what would be like the apex of a mountain or a tree? Would be the top, right? That's it's usually the best, the top, right? So it's the top of the tree or the end of the growing piece, whatever that may be, and also the end of the root. Wherever the end of the growing piece is, that's your apical portion. So your apical meristems, those can obviously differentiate as well. That's how you get it to keep growing up into other tissues. And then let's see, the unspecialized cells that result from cell division meristems have thin walls. Newly produced plant cells undergo differentiation as they mature into different cell types. And then lastly, apical meristem changes into floral meristem. When its pattern of gene expression changes. So plants are useful for many fibers such as cotton and linen. Fibers are long, thin structures that have strength and flexibility. Which plant tissue produces fibers such as cotton? Do we know? Otto. So plant fibers such as cotton and linen are produced by the vascular systems of plants. Vascular systems contain long, slender cells that form long tubes and have walls that are reinforced by ligand. Ligand is the stuff in trees that makes wood hard, right? It's fibrous, it's very fibrous. And these characteristics of those fibers, such as cotton and linen, are similar, sorry. And then neither dermal nor ground tissue consist of cells with their characteristics. So it's vascular tissue that produces all the cotton fibers, that kind of the same type of tissue. So we did stems, now we're doing roots, all right? Roots, types of roots. We have different types of roots. Tap roots and fibrous roots. So your tap root is a thick primary root, grows long, and it's found in a lot of dicots. It's also found in a lot of conifers. Have you noticed that pine trees tend to fall over when there's big storms? They tend to be the ones that rip up. Have you ever seen what it looks like when you look at their root system? They have a big tap root like that. Their roots go down like this one here. They don't spread out quite as much. You've seen an oak tree roots? They go this way, right? They're all spread out. So any of these things with tap roots like this is the thing that in your yard, remember, I think we talked about it last week, but if you pop this dandelion off at the top, you have not killed it because its root goes way down deep. My grandfather used to walk around with a screwdriver, pop them out of the ground. You have to get the whole root out. So you have tap roots, and then you have fibrous roots. And these are the kind that branch extensively, and they're more commonly found mainly in monocots. So you got dicots and monocots, and their root systems can be a little different there. All right, now roots, structure and growth. This is a really good graphic of a root. I spent a long time trying to find one that had everything in it. So root hairs, this is like the one thing it didn't have, are these tiny projections from the epidermis, right? They increase the surface area. Explain how's that work? How's a little hair increase the surface area of the root?
because it's a cylinder, so all
Mhm.
sides of it.
Right? All sides of it plus the root itself. So the more little root hairs the root produces, the more surface area it has, because each one of those all the way around it is more surface area, right? So all that surface area is exposed to water and nutrients. So it's beneficial to produce those. Now you got this cortex, all right? Cortex is your layer of ground tissue. This is this right here. So it's in between your dermal tissue and your vascular tissue. You've got this stuff, that's your ground tissue, right? And then you have in a root what we call an endodermis. So it's a layer of tissue that encloses the vascular tissue. So it's right in here, right, this little green thing here, all right? So this is keeping your vascular tissue protected, all right? Think about what vascular tissue is doing in roots. It's doing a couple things. Obviously, it's doing the obvious thing. It's sucking up water and nutrients, right? But it also has to be rigid and firm enough that it really roots the tree in the ground, right? You don't want your tree or your plant or whatever to blow over. So it's part of really grounding it so it doesn't get knocked down or whatnot. And then you got your root cap. That's this little thing here. And it protects the root as it forces its way through the soil. So this root is full of all of these structures that are super important. And it has to push through soil and run into rocks and sand and goodness knows what else. So this little root cap is kind of like, you can think of it almost like a fingernail, right? Like if your fingernails protect the tips of your fingers, right? It's the same kind of thought, right? Or it's like a steel toe on a boot, right? It protects that root as it pushes through. So root function, anchoring, right? This is what we were just saying. It really anchors to the ground. and then water absorption and dissolved nutrients. So if you look here, it's got an example. So what do we see? You see this root is moving around things. What is that? What tropism are we seeing? Do we know? It's a TH. It's thigmotropism, right?
Oh, is that? I thought it was
Thigmotropism. So this root is moving along and it's growing in a direction and it gets right here and it goes, uh-oh, I can't move around this little rock or whatever it is. So what does it do? It has to turn. Now it has a choice. It can turn up or it can turn down, but roots tend to turn which way? Down, right? So it gets here, it needs to turn down. So what hormone comes in? Remember? Auxin. Auxin comes in and then what does it do? It tells it what?
Go down. It tells it where gravity is.
Yeah, what's it do? And how does it do it?
It changes. It tells the cells to grow long.
Tells the cells on which side? The side that's touching, in this case, to grow how? Longer, right? So then what does that cause? That causes it to, if the cells on this side are going to grow longer, it's going to bend away, which is exactly how it works, right? And it's done it several times here, as you can see. So when you pull roots up and you see them looking like this, they always look like this. That's why they're not nice and straight, because they run into stuff the whole time. All right, so for this one, let's see, you had tap roots and that they're more common in dicots, and it gave you examples of carrots, dandelions, beets, and then your fibrous roots are more common in your monocots. All right, so mature root has a large area of ground tissue between dermal and vascular tissues. A root surface for absorption is increased by its root hairs, which we just talked about. The cortex is for storage of starch, and it protects all your vascular tissue in the middle. And the vascular cylinder is made up of xylem and phloem in a root. And then this apical meristem can be found just behind the root cap. There's your apical meristem. That's what's growing, right? And then your root cap is protecting it, because that's super important. So two functions besides water uptake and nutrients of a plant's roots. So what are some functions? So aside from the obvious, I also said roots are there to do what?
They anchor.
They anchor, right?
Yeah.
And what else can they do? Storing food, right? Absolutely. So the role of active transport in the uptake of water in plant roots is energy from ATP is used to pump mineral ions of dissolved nutrients from the soil into the plant. And that increases the concentration of mineral ions in the root cells. And as a result, water moves into the plant by osmosis. Okay, just like, think of those poor slugs again, right? If you pour salt on a slug, what's going to happen? It's trying to find homeostasis. It's all homeostasis for all living things, right? That's what everything wants. Homeostasis is balance. Remember, balance between the outside and inside. So if you've got more ions outside than inside, they're going to move. And if you've got more ions inside than outside, the water, you're going to get water moving in and out of the cell, trying to balance. That's always the way things go. So what else was important here? What happens to water and dissolved minerals after they move across the epidermis of a root? The water and dissolved minerals move from the epidermis through the cortex and into the vascular cylinder. And then where do they go? Wherever they're needed, right? Wherever they're needed. Why is there a one-way passage of materials into the vascular cylinder in root plants? Because water and minerals cannot pass through waxy Casparian strip, another part of this thing, right? Once they pass through the endodermis. And then root pressure is what forces it to move on. So people often give potted houseplants more fertilizer than they need. I'm reminded immediately of Little Shop of Horrors, where he talks about all the things that he tried to give that plant to make it grow. As a result, the plants begin to wilt and eventually die instead of getting larger and healthier. So why is that? Why can giving it too much nutrients kill it?
Osmosis.
There you go, osmosis. What's going to happen if you put too many?
There's too many nutrients in a deficit of water.
It's gonna lose water.
That's right. Exactly right. Exactly right. All right. So now we're onto stems. Stem types. Oh boy. We have two types of stems, one from your monocots and one from your dicots. And this is the cool thing about monocots and dicots. If you have a seed, it's really easy to identify if it's a monocot or a dicot. It's like super easy. You look at it, you go, oh, obviously, I can split it into two pieces. It's a dicot, right? But you can also tell monocot versus dicot without the seed. Because remember, you can count the parts of the flower and see if they're in multiples of 3 or in multiples of 5 and 6. You can also look at the leaves and see whether they have branch veins or parallel veins. And you can also look at the stems if you get a cross section. So that's what this is. It's a cross section. So your monocot has these vascular bundles that contain xylem and phloem, and they're randomly scattered all throughout it like this. But in your dicots, they're really nicely organized like this, right? So they have these vascular bundles, but they're in a ring-like pattern. So if you see something with a ring-like pattern, like think about an oak tree. If you cut an oak tree, you're going to see this repeated many times, right? All the rings. Exactly. So what's the function of a stem? Why do they have stems? I mean, some of it's obvious, right?
Transfer just to be strength to grow up.
Structure.
Yeah, transport and structure. So they produce the leaves and the flowers. Come on, baby. Are you coming to me? Come. No, you're not? Okay. They produce the leaves, the branches, and the flowers, obviously. And they hold the leaves up to the sunlight. Now, this is important. The leaves are your primary photosynthetic organs, right? So although you've got chloroplasts in the stem of a plant, your leaf is where you're doing all of your energy production. Energy, what do we want to call it? Transfer of energy from the sun through the chlorophyll into sucrose, right? That's where your photosynthesis is taking place. So without a stem, you have to get the leaf up to the sun. That's why they bend, right? So transport substances between the roots and the leaves, obviously. We were talking about that. So you got different kinds of stem structures. Stems have structures. So we have nodes, and that's where the leaves are attached. You can see these like really obviously in some plants. You go and you look and you see the little, like they're little nodes or little notches. And you go, their leaf is going to come out here, right? And then you've got internodes. So these are the regions between leaves. This area right here is called the internode, okay? And then you have buds. There's a bud. You all know what buds are. So they contain undeveloped tissue. So sometimes buds aren't going to produce flowers. They may produce more stems, more leaves. So in this case, this one's probably going to kind of take over this leaf. This leaf may get just squashed out of the way as this thing grows. So this one's cool because it really breaks down this stem. This is a little woody stem, and it's got cross sections for you. It's got a longitudinal section right here. Yeah.
Why does the top look like asparagus?
I think it's a bud. And these are little buds here. But, and these would be nodes probably, and then your internode space is here. But I liked it in that it gave you a really good longitudinal section, and then it gave you these cross sections. So, wait, didn't mean to click it yet. So I was gonna say, you can see here, you've got your different layers, right? And you've got this layer of what they're calling cork. This is your bark, right, basically. And then you've got your layers of your cambium and your xylem a lot of the time. You're gonna see where it builds every year. That's how you get, we talked about tree rings, right? And how you can continue to watch them grow. All right, so what are your three main functions of stems? Hold up the plant, transport materials, and then get the leaves to the sunlight, right? Those are your big ones. So what's an example of a stem that conducts photosynthesis and stores water?
Cactus.
Cactus, right? So a desert cactus stem conducts photosynthesis and stores water. All right, if you're stuck in the woods by yourself and you have no water, right? What about a cactus tells you that you could probably get water from it? Do you know, like looking at it,
It's pretty big.
it's big, right? It's very big. Cactus is all full. Yeah. Why don't you want to touch a cactus?
Well,
It's got thorns on it, right? Okay. Why does it have the spines or thorns? What's the point?
Because it wants to keep its water.
It wants to keep its water, right? So a lot of the times when you look at plants, a lot of the times the plants with thorns are actually the edible ones. Think about blackberries, raspberries. It's not always true, but a lot of the time the edible plants are the ones with protective elements. Like, don't eat me, I'm covered in thorns. But you know, a cactus has got lots of water in it, tons of moisture in it, right? So we talked about nodes. That's where the leaves grow out of the stems. And we talked about buds being where you've got new stuff growing. So what kind of tissue is in there? What kind of cells are you going to find in a bud? Meristem cells, right? Meristem stem cells, which in plants are meristem cells. So let's see. So you got your monocot, dicot.
What's the root for Mary?
You know, I don't know. That's a good one. Let me see what it says. Because stem cells, we know.
Etymology of meristem.
Yeah, it's a prefix in this case. So let's see. That's a good one. I'm not sure. Let's see. Yeah, I love etymology. I agree. Prefix merit. And that's merit. Uh-huh. Merit. I keep getting merit. That may be as close as we're going to get. Words derived from merit. Oh, Latin. Merit, mere. A quality or characteristic deserving of blame. No. Absence. Mere. Oh, Latin. To deserve, to earn, to acquire, to gain. Maybe to gain or to acquire. Growth. I'm not sure. It's interesting though. Somebody look up the etymology of the word meristem. We'll see what we get.
Is it etymology?
Etymology, and that was it?
You said
Entomology is the study of food sex.
What I say? Did I say etymology or
No,
entomology?
I said etymology.
Oh, etymology is
And
what I
then you
meant.
said ed.
I threw in an N that doesn't belong. Etymology. Yep.
Okay.
All right. So your monocot and dicot stems both have your vascular tissue in them, and they have the same tissues, but the structure is different in that your monocots have their vascular bundles all spread out, and your dicot have them arranged in circles. All right, so plants grow in a way that is the same way animals grow. Is that true or false? That's obviously false in a different way. So the number of legs an animal will have is predetermined, but the number of branches a plant will have is not predetermined. Is that true or false? That's true, right? Primary growth of tissue is the result of elongation cells produced by what? Not ground tissue, but apical meristem. The increasing thickness of stems and roots and dicots and gymnosperms is called secondary growth. So if you remember in the Professor Dave video, this was primary growth, this was secondary growth, right? Primary growth, secondary growth. And it's kind of funny, humans sort of do the same thing. If you watch kids, especially like kind of prepubescent kids, you'll see them kind of widen. I don't know. It's like they gain weight suddenly. They become a little wider and then they shoot up. Like right before a growth spurt, you'll see them kind of widen and go up and out, up and out. Plants do the same thing. It's just way more obvious, right? So secondary growth is rare in monocots. Dicots can grow to great heights because the increase in width supports their height. That's true. Vascular cambium forms between xylem and phloem in vascular bundles. Yes. In conifers and dicots, secondary growth takes place in the meristem. And we talked about outer coverings and leaves and all that good stuff. So here's a nice little diagram for you. Is this a monocot or a dicot in this picture right here? It's dicot. How do you know?
Regularity.
Because it's in rings, right? Absolutely. So most of what we call wood is made up of layers of secondary xylem. The dark wood that no longer conducts water is called heartwood. Okay, if you're looking to burn something, people really like heartwood for fires. Actually, really weird story. I'm trying to remember what he called it. At our old house, we had woods behind our old house where they were building a neighborhood, but there was also some houses back there that had been abandoned. And the people that originally owned it had a house way down the road that wasn't abandoned, that they were in. And there were a couple of like ATV tracks through the woods. So one night we were down by our fire pit down by the creek, and this random dude with a machete comes walking through the woods and crosses the creek into our yard. And I was freaked out. I'm like, what is this dude doing? And he starts commenting on our fire. And then he told us we needed, oh, what did he call it? It's another word for heartwood. He called it, but anyway, and then he wanders off into the woods in the dark and came back with some. And I was like, dude, what are you doing? He scared me to death. I thought we were all going to die. Like, he's got a machete. It's dark. He's wandering around the woods. Who does this? Like, who does this?
Here's getting some backwood.
Yeah.
On the bright side, now you have a great story to tell.
And I had no idea what he's talking about.
You want to know the time I almost died in the
I can't
woods?
remember what it's called. I can't remember what it's called. I'll have to keep looking. But anyway, people, oh, fat larder. That's what he called it. You need some fat larder. And I'm like, what? So anyway, you may run into that, but that's the heartwood. Okay.
I mean, I hope I don't.
It's down in the center and it no longer conducts water. So it's very dry, which is wanted it. It's also a lot of the times when you see trees decay and they leave behind sort of like an inner structure that's kind of bendy and neat looking, that tends to be the same structure. I have a couple that are out there kind of in our little garden area because they're pretty. So let's see, wood that is active in fluid transport is what you call sapwood. And the lighter wood in the tree rings contains larger cells with thin cell walls. Alternating layers of light wood and dark wood are used to estimate a tree's age. And then here's a good cross section of a tree. So girdling is a term that refers to removing the bark of a tree in a complete ring around the trunk or the branch. So what's the effect on the tree? What would that do? Nothing good.
It would make it vulnerable to diseases.
Right, because it'll eventually die. And this happens because the phloem vessels that conduct products of photosynthesis to other parts of the tree, including the roots, are cut and no fluids are able to flow. And also it opens it up to all kinds of diseases. This is why you need to leave bark on trees. Like some people start pulling bark off trees or whatever, messing with it and, nailing stuff into it. You have to be very careful because it's like opening up your skin, right? You open it up to whatever's out there. So leaves have structures. These are the structures of a leaf. You probably remember this word, petiole. Remember when we did when we measured the leaves and we found the average lengths and all that? That's the petiole there. So your leaf blade is this kind of the main part of the leaf, the big flat part of the leaf, right? Your petiole is the stalk that attaches the leaf to the stem. And then you've got veins, and that's xylem and phloem. And remember, is this going to be a monocot or a dicot? Can you tell by looking at it? That one's going to be a dicot because it's got branches. If they all ran straight like this, like a corn or grass, that's your monocot. Oop, that was the name of that one. I thought there was more. So then you've got different kinds of cells within your leaf. So here's this guy again, right? And what's this right here? That's your stoma, right? We saw these last week, and it's a good close-up of your guard cells, right? So your leaves have your palisade mesophyll, and these are these tightly packed ones here along the outside. You've got your spongy mesophyll in the middle, and then you've got your stomata and your guard cells. Remember, these guard cells open and close, right? And they're doing that using what, do you imagine?
Vacuums.
Yep, vacuoles with water, all that same kind of thing. And remember, this is the same way that we saw some of our protists move, right? They used pseudopods, for example. Yeah, the amoebas did. And a lot of that is, again, where your vacuole, what's going on with the vacuoles and where they're pushing their cytoplasm from one place to another. So this is all, if you think about it, you can see the evolutionary movement from one type of organism to another, right? You can see how it goes. So then we have our photosynthesis, which is the primary function of our leaves, right? Got to have that. Takes place in the mesophyll. That's the part. So you got to go through the palisade cells. That's your mesophyll, right? And then what's transpiration? That's your water loss through your leaf. So leaves undergo photosynthesis and also transpiration. And so when you have, you ever had like you ever seen those little terrariums? And they can get foggy, like condensation on the glass or whatever. That's again because transpiration. Those leaves are releasing excess water into the air, and that, depending on the external air outside the terrarium, it might form a nice little fog there. And when you lose water in your leaves, what happens is now you have less water in your plant. So what's it going to do? It's just like a straw. So it's almost like if you suck out of the top of the straw, what's going to happen to the bottom of the straw? Water's going to come in through the roots. So the purpose of transpiration is to form almost like a vacuum from the bottom to the top of the plant and keep sucking the water upward, because your leaves are at the tippy top and your roots are at the very bottom. So it's moving water up by losing it. And then you've got gas exchange. Your carbon dioxide is coming in and your oxygen's going out, obviously. So for these, let's see. Leaf structures optimized, obviously, for photosynthesis. Your epidermis is covered by a cuticle. We've talked about that. Where's one that we didn't talk about? Okay, a layer of mesophyll cells that absorb light that enters the leaf. These are your palisade, right? Your small openings are going to be your stomata. Your thin, flattened part of your leaf is going to be the blade, and your bundle of xylem and phloem tissues in a leaf. What's that? That's your vein, right? Stalk that attaches to a stem, that's your petiole, and your loose tissue, That's going to be your spongy mesophyll. So why can't stomata be open all the time?
Water loss.
Yeah, too much water loss. Absolutely. You also have weird gas exchange too. So complete this flow chart. So it starts with guard cells are forced into a curved shape when water pressure does what?
Rises.
Yeah, when it increases, all right, you're getting too much water pressure, your guard cells are going to go, and that means that now your stomata is open, right? The thick inner walls of the guard cells pull away from one another, opening the stomata. So then you lose water by transpiration. Your guard cells straighten out when your water pressure decreases. So your inner walls of the guard cells pull together and that closes it. This makes absolute perfect sense. If you have too much water in your plant and you need to lose water, okay, you've got too much water in your plant, what's going to happen to those cells? Guard cells are shaped so that they bend. The more water you put in, they're going to bend because now they're forced into that shape. When they lose water, they're going to straighten out and they're going to close. So you're going to get an open stomata and a closed stomata based on your water content of your whole plant, basically. So which is likely to happen if if it starts losing more water than it can take in?
Yeah, it's gonna die.
Yeah, it starts to wilt, really flops over and looks awful. So which is a plant that has narrow leaves with a waxy epidermis? They have cactus, spruce, rock plant, or rose bush. So if we're talking about narrow leaves, we're going to talk about spruce. Yeah. What's a spruce look like?
Pointy leaves.
Yeah, like a pine. It's a pine tree. It's a conifer, not a pine tree specifically, but okay. Pitcher plants leaves are adapted for what?
Eaten.
Yeah, what does it eat?
Insects.
Insects.
Frogs.
Yeah, frogs. So pitcher
Whatever
plants.
falls in.
Yeah, pitcher plants are one of these weird carnivorous plants that can get additional nutrients from other organisms that it catches, from animal organisms that it catches, like a Venus flytrap will do that. But these pitcher plants, you've seen them, right? They're big and open, and then they have these, it's either sticky or slippery, and it causes little critters to get sucked in. Yeah, sometimes entire frogs, which is amazing. So a rock plant adapts to hot, dry conditions by having very few stomata, and cactus thorns are actually, it's what?
Leaves.
There's leaves, right? Very good. So the inside of glass or plastic walls of a greenhouse, like we were talking about terrariums, right, is full of plants in very wet, a greenhouse full of plants is very wet on cool days. So where's the water coming from? It's inside the walls of the greenhouse are wet because water vapor from air condensing on the cool glass. The water vapor from the air comes from plants through, what's the process where plants release water? Transpiration. That's the one. So now we talk about transport in plants. So water transport, where's that happening? What tissue? In xylem, right? Very good. So transpiration is the technical term for evaporation of water from plants. As water evaporates through the stomata in the leaves or any part of the plant that's exposed to the air, it creates a negative pressure, and we call that tension or suction in the leaves and the tissues of the xylem. So here's your water, it's traveling up by the roots, water leaves, you've got negative pressure. What does that cause? It's just like a straw. What happens down here? It brings the water in. It's just like a straw. You can just imagine that instead of sucking into the straw, it's getting sucked into the atmosphere around it, right? Whoop, sorry, wrong button. So the negative pressure exerts a pulling force on the water in the plant xylem and draws the water upward, just like the water upward when you draws upward when you suck from a straw. It's the exact process here. So you get water from the roots, it goes up, it evaporates. It's this cycle that keeps pulling it up, pulling it up, pulling it up. So plant cohesion, what's this? All right, so 3 forces transport water in plants. So we just talked about one. So here's cohesion. When water molecules stick together through cohesion, they fill the column in the xylem and act like a huge single water molecule, like water in a straw. So think about, bless you. Think about capillary action. Do you guys remember what capillary action is?
I do.
What's capillary action?
It's where like water moves up really thin tubes.
Like what's an example of capillary action?
Plants.
Yeah.
Those pooled pipe things being
Yeah.
Paper towels.
Paper towels is a good example.
Oh, yeah.
You ever go to the doctor and they take that tiny little glass tube after they poke your finger and they touch it to the little drop of blood and it goes, whoosh, right? Okay, that's cohesion, right? That's the water molecules there are stuck to each other. Because remember, water molecules love to stick to each other. They do. And so it's going to act like one big unit. And that's part of what's helping the water move through the plant so effectively. So this is an example of capillary action right here. So these tubes are different diameters. And if you stick them all in water like this, if you stick them in a dish of water, you're going to see that the thinnest tube is going to suck up the furthest. Okay. It's not necessarily that the volume is more, but we transported a greater distance. And
The
that's
volume
due to
is
cohesion.
less. I mean, the volume may be equal.
It might maybe equal, volume may be less, but we're talking about distance and transport. So capillary action, here it is the movement of liquid across the surface of a solid caused by adhesion between the two. So the water is sticking to the sides here, and there's more, over here, there's more surface area, so it's kind of harder for it to attach. But here, it's just going to attach and go straight up, right? So when you place a tube in water, water automatically moves up the sides of the tube because of adhesion, even before you apply any sucking force. The narrower the tube, the higher the water climbs on its own. In plants, adhesion forces water up the columns of cells in the xylem and through the tubes in the cell walls. So thank goodness for that. Nutrient transport. All right. How do we get sugar from one place to another? How do we get minerals from one place to another? Changes in nutrient concentration, drive the movement of fluid through phloem tissue directions that meet the nutritional needs of a plant. So remember, your phloem is moving things like sucrose from one place to another. And we're going to call that translocation when we're moving something from one place to another. And it's all about the concentration. So the leading explanation, here we go. The leading explanations, now we're in theories, right, of phloem transport is known as pressure flow hypothesis. And that's what's shown here in this figure. So what's going on here? We have this little tube here and this little tube here is your xylem. It's sucking up water. Here's your phloem. Somewhere in here, we're seeing there is more sugar over here than over here. So what's going to happen? We're going to move more water into here so that we've diluted it, right? Remember?
Yeah.
All right, so the membranes of sieve cells. These sieve cells again. These are the little guys over here. And these are actually them, even though you can't really see them. Can use active transport to move sugars from their cytoplasm into the sieve tube itself. Water then follows by osmosis, always osmosis. That creates pressure in the tube at the source of sugars. And then this last little bit says, if another region of a plant has a need for sugars, they're actively pumped out of the tube into the surrounding tissue. Water then leaves the tube by osmosis, reducing the pressure. The result of the pressure flow system is the flow of nutrient-rich fluid from sources of sugars, which are source cells, to the place where the sugars are needed or stored, which are called sink cells. That's down here. So it's getting it from here and it's moving it to here. What you need to remember is that in nutrient transport, the phloem is involved, and it has to do with concentration gradient, and it has to do with osmosis. Osmosis is what fixes your concentration gradient. So this pressure flow system gives plants flexibility in responding to changing seasons. So during the growing season, sugars from the leaves are directed into ripening fruits or roots for storage. So your plant in the summer has all these leaves and it's doing all this work, but look at the deciduous plants out there right now. Are they making any food right now? They got no leaves, so they're not making any food. So what happens to it? It has to get stored somewhere to get it through the winter. So as that growing season ends, the plant is going to drop its roots and store its nutrients in the roots. This is where your ethylene comes in, by the way. You're going to start pumping in some ethylene, and it's going to cause the flowers to fall off, the roots to die. The leaves to die, and it's going to start pushing your nutrients into the roots. So when spring approaches, the phloem cells in the roots start pumping the sugars back into the phloem sap, and the pressure flow system raises these sugars into the stems and leaves, and then starts growing again. Ooh, and one more thing. In this image, you can see these are what? These are potatoes. Yeah. So, or any other kind of tuber. When you get a tuber, a potato is a tuber, right? And it's a sink. This is where we put all the extra stuff. So it's sucrose, and it's been converted into starch. So when you eat a potato, right, you know, it's like all carbs, right? It's all starch. So I've taken the sugars, converted them into starch, which is a really great way to store stuff, and stuck it down here, right? Makes it super easy for us to get. So here's an analogy here. It says visual analogy of the climbing circus clown. Ah, I love it. Is what makes it possible for the falling clowns to pull others up the ladder. So the rope that ties the clowns to each other transfers the force of gravity, pulling the falling clowns back through the chain and still climbing. So this is your water movement through your plant, right? Clowns. I don't know why they chose clowns. They're trying to scare people. So how are the water molecules similar to the clowns? Great question. So the force between the water molecules is keeping them together. There's your cohesion, right? Transferring the pull of transpiration back through the rising column of water. All right. So cohesion versus adhesion. Cohesion are your water molecules sticking together. Adhesion are your water molecules sticking to the outsides of the tube. And you need both of these things to get capillary action, right? They both have to occur. Water cohesion is especially strong because water molecules form hydrogen bonds. We've talked about this before early on, and then we talked about it a lot in chemistry. So water molecules have hydrogen bonds that keep them stuck together, right? And then when you move water through a tube, that's our capillary action that we've talked about. So the height to which water can rise is determined by the tube's what? How high the water can rise is determined by what? Yep, diameter of the tube, right? Diameter. Yep, absolutely. So your big ones are not going to do as much. Your little ones are going to do a lot. All right, let's say transpiration extends from the leaves to the roots. All right, according to, this is this thing we just talked about, where sugar concentration is high. What is the sucrose of water taken in by the phloem? Water drawn in by osmosis in areas of higher. That's a terribly worded question. Sorry about that one. How does the structure of vascular bundles in stems and roots and in the veins of leaves make the process of pressure flow possible? So these water bundles in the veins of leaves contain xylem and phloem, right? And they're close together, which means that water can move from the xylem to the phloem without having to pass through too many cells. So that's how you get all that activity in the leaves, water coming in, nutrients going out, flowing around in a big cycle. So you guys have this week's lesson, and then you have next week's lesson, and that's the end of plants. And then we're going to move into animals. And I mentioned before, you're going to want to look ahead at your labs. If you're not going to want to do a lab because you're like, ooh, I can't dissect whatever, then you need to look at the alternate lab. And in those cases, the alternate labs need to be printed by you. They're all available in Teacher Ease. They're all there, each one. You need to be able to print them. And some of them will say, do this part and this part at home, and then come to class prepared. So you're gonna wanna look at it ahead of time. You're not gonna wanna say the morning of, oops, I'm gonna print this, because some of the work you're gonna need to do before you get here, if you're doing the alternate labs. Some of the work you're just going to do once you're here. And what we'll do is we'll put you over in the pub so that you can work over there and not have to watch whatever being fun. Does anybody think they're going to want to do the alternates for some of these, for any of these? Or is everybody good to chop stuff up? I want to try and chop stuff up. Yeah. As long as there's, are we needle phobic? So it's not really, I don't think any of it's too bad. I mean, you may not like, you may not like the well, I was gonna say the pig gets a little complex. Maybe you don't like the pig, but yeah, so the pig looks really human. And it does, because I mean, we use pig tissue for certain things for humans. Yeah, we've done heart valves, pig heart valves. Yeah, because they're very similar to ours. So we've done that. Yeah, very cute, right? Yummy, delicious heart valves. So it starts with this, next week, we do have our first dissection, but it's a flower. We're dissecting a flower. So that's not anything that's going to freak anybody out. But we're going to really look closely at all the different parts of the flower, and we're going to work our way through it. And we're going to just complete that in class. That should be easy. And I'll probably for the flowers and for the roots, I'll do one per team because that's easy enough. But we're gonna move into when we move into the animals, our first is gonna be this worm, and then the sea urchins. Sorry, I couldn't remember the name of them. Like those things, what are those things called? So when we get into the sea urchin, that one you're gonna like, it's gonna get dissolved in bleach, like I said, and you're gonna be able to take it home probably, and you're gonna have to clean it up and bring it back the next week. So it's smelly, but it's not too terrible. And you probably wanna take the spines of it, like you probably wanna do it outside with a bucket. That's my best recommendation, rather than in your house, because your mother will probably be like, no, oh no. And so your worm, if you haven't already looked, I just want to give you an idea of what you're looking at. Okay. When you get into your worm dissection, this is what you're going to be, open up. This is what you're going to see. Most of these labs do have pictures. Some of them have actual pictures. Okay. So that's helpful. So when you get into your worm, you're going to see something like that. This one has a diagram, not an actual picture. Some of these have actual pictures to tell you exactly what you're going to see. So it's going to look like that in your worm. It's not the end of the world. That one's pretty easy, right? Just a worm. Just a worm. You've done a worm before? Worms are easy. Okay. When the next week, we're going to do sea stars, which are, starfish, sea stars, and the fish as well. So the sea stars, you're really just going to kind of really look at it, flip it around, study all its parts like this. There really isn't like a cut it open too much kind of a thing. You can, so if you want to see what's inside their arms, which is pretty cool actually. But mostly what you're looking at is their exterior structures. When you finish the sea star, you move into the, where'd it go? It's actually a perch, correct. So here's your sea stars again, whoop, whoop, whoop. Yeah, perch are edible. When you, and there's a squid too, actually. Actually, it's the squid, not the perch. That's the next week. So you finish up this and then you get to the squid. Okay. Squids are going to be stinky. All right. All these are going to be somewhat stinky. Yeah. I don't know. It's kind of cold this time of year, but maybe we might. It depends if my husband freaks out about it. If he doesn't, we'll do it inside. But when you do your squid, okay, here's a picture of the squid. Come on, get to the picture. Here's your outside of the squid. Oh, this one doesn't have a good picture. So you'll just be looking for the inside parts and it'll tell you, this one has more of a description of what they look like. When the next week comes though, this is the fish. Oh, no, not 26 yet. Hold on. 25 is the fish and the frog. Where is it? Alternate key. There it is. For this one, you're going to be looking at a fish first. There's going to be goldfish for you to observe. You're just going to observe them. Cool, it's a goldfish. Wow. Anybody wants to take them home? Fine. Otherwise, I'm just going to hang on to them until they croak. They will be live. Yep. They will be live goldfish swimming around. You have a tank. It'll be your little tank. You're going to go catch. There's a big tank. You're going to catch the fish out of the big tank. You're going to put it in your little tank. You're going to watch.
Is it going to be a hammer test like at the fairs?
I know. You're going to watch your fish and observe it. All right. Then you're going to get, then you're going to move on to getting your perch. Okay. When you get your perch here, they're going to really stink probably. And you're going to dissect your perch for sure. I mean, you're also going to look at the scales. It wants everybody to have a microscope to look at the scales. I'm probably going to have one microscope set up with the scales so that people can just come by and look. So I don't have fish hands on all the microscopes because that's a bit much. I don't mind cleaning them, but oh my goodness, that's a bit much. So you're going to look at your outside of your fish and you're going to be looking at the inside of your fish. And then you're going to do your frog. Okay, it's going to be a busy day. When you get into your frog here, it's going to be the kind of thing where you're going to be running. You're going to be doing them quickly. Like looking at the fish isn't going to be hard. Dissecting the fish, we have, labs an hour and 15. So there's time to do it. With the fish and the frog, the way things will go with these is I will have like I said, I have the little pins that are numbered. And you'll be, for the pig, you'll be popping them in. For this, you'll just be starting to look at things and pointing them out and going, okay, I found this and I found that. And that's important because when you get to the pig, you're going to need to be able to show me everything and have it right. So that one's going to be, and some of those organs are going to be similar, like a liver in a pig and a liver in a frog are not that different. Mhm.
Are some of these, are they gonna be like lab, like all the labs, like some return and some? keep
Yeah,
binding,
most
or-
of these are going to be keep in binder, but the pig one is a big one. So the pig one you're going to really get graded for, and you're going to have to, like I said, put the little pins in. So your frog, when you get him open, will look kind of like that.
Yum.
Mhm.
Are we talking like small?
Their frogs are about this big.
I'm
Mhm.
like, that's a bigger pot really. just
They're-
put it on the counter, right?
Double injected. Now the pigs are about this size. They're good sized pigs. You know, they're fetal pigs. So they're always fetal pigs. That's what you use for dissections because they're smaller. Sounds atrocious, I know. Lastly, this is the one that some of you may be like, okay, because it's a pig. All right. So what you're going to be doing is this the one where I have it numbered? I think this is the one where I have it numbered. Yep. Yeah, this is it. Okay. This is how it's going to work. So there is a whole thing here about why fetal pigs are great. You're going to measure your pig. You're going to sex your pig. By measuring your pig, you'll be able to tell how many weeks gestation it is because it is a fetal pig. You're going to sex your pig. Oh boy, is it a boy or a girl pig? And then you're going to check his mouth first. And this is an actual photo. This is what you're going to see, right? You're going to open it up. And the way that you open their mouth wide enough to tell is you're going to have to cut them this way so that you can really get inside their mouth and see what's going on in there. Mhm.
I was about to say like, I'm the supervisor.
Yep. And then you have these various, various incisions that you're going to make into the pig so that you can cut him open all the way and see all of his good stuff inside.
That's like nightmare fuel for some people.
That's what I'm saying. When you get inside the pig, this is what you're going to see, actual photo. Now, you're going to take the pin labeled number 1 and you're going to stick it in the liver. And you're going to take the pin labeled number 2 and you're going to stick it in the stomach. And this is how you're going to get graded for the pig. Okay.
Has
How
it
many
contained
posts is the
anything?
pig worth?
These were the full lab. This is
Okay.
kind of, it's usually, this is like the big lab practical is your pig or your cat. It's whatever your mammal is typically. Cats are great. And I would love to get cats, but our budget didn't allow for it. Cats are fabulous because they're bigger and you can really see what you're doing.
Dad had Calvin.
Yeah.
All the cats.
And so you can see the body parts here with each of these little pins. This is where you're going to stick these little pins in. I put them in here so you can see where they go. It's like example, your gallbladder is up underneath your liver. You're going to want to find it in there. You're going to stick pin number 9 in it. I found it. And then you're going to leave them. Like you're going to get all your pins in and you're going to leave them for me. And then I'm going to go by and grade them all. Like for real. You got your urogenital system. So if you have a male pig, you're going to stick number 10 in its scrotal sac and number 11 in its epididymis, right? Number 12 in its vas deferens. You're going to stick it. You stick number 13 in his penis and number 14 in his urethra. And then the same thing for the girl pig. If you have a girl pig, you're going to stick pins in all the girl pig parts. So I know that you know where the uterine horns are or the oviducts or pig vagina. So there it is. You'll be pinning all kinds of stuff. And this shows you male versus female pig, right? So you're going to be looking at that. And when you get to the thoracic cavity, you're going to do the same thing, heart, lungs, esophagus, larynx, all this stuff. You're going to be pinning the heck out of your pig, 20 pins. And it says, place this pin at the top left corner of your tray and label your pig male or female along with gestational age. This is the one that tells me you're done. Yes.
So are they already gonna be like open? Do we have to open them
No,
up?
you got to open them. They're coming.
Like that's like a full off. That picture up there
Mhm.
is like off of it. So we gotta.
Yep, they're gonna come looking like a pig. You got to do all the opening. Now these are double injected, which means they've had latex shot into their arteries and their veins. Their arteries will be pinkish red, their veins will be blue. You can tell one from the other. It's really cool. There's no blood in them. They've been drained of blood and they've been put in, a preservative fluid. So they're going to be pretty stiff usually because they're preserved. Yeah, and when you do, like, for example, what you're going to do here to get it in this position, this is not an easy position to get the pig in. So because they're stuck like this in the bag they come in, so you tie string around them, you loop it around the back and you crank them open. I mean, you crank them open And you tie him down, like you can tie it under the tray even, and it will hold him out like he's, I don't know, going to be drawn and quartered. Basically, he or she will be drawn and quartered. And the cool thing about the fetal pigs is they do have an umbilical cord, right? If you had a completely mature born animal, you wouldn't have that. But you can definitely see how this attaches into the rest of the body, which is the same way it attaches with all vertebrates. It's pretty cool.
Which lab is this?
This is lab 26,
Oh, so
I
it's
think.
16.
It's in a couple weeks. This is, yes, 26. This is the last dissection.
In a month.
Mhm.
a little more.
Yeah, it's on the 26th, will be on March 8th. So you will have had, we'll already had fall break because that's February 22nd. We come back on March 1st, and then on March 8th, we're doing the piggy. Yes,
We'll have fall break in February.
yeah, not fall break. What did I call it? I said fall break, but it's winter break. Sorry, my mouth just doesn't work. I have all kinds of new meds in me. And it says completing class and keep in binder because you are going to keep your lab in your binder, but your team's going to leave one for me. Your team's going to leave the pins for me and one little thing that says whether it's male or female and its age. So you'll be able to keep the lab with you, but you're going to leave your results for me. Like you're going to leave me the pig. And most of these, like I'm saying, you're going to do it, like it says, complete lab, due for hand in. So you're going to take the fish one home and actually hand it in because that one's a little more complex. No, the lab home. But most of these are like completing class for your, this is the one for your
We take a fish home?
starfish and stuff. You're going to take those goldfish home. I don't care. So, and the same thing with the worm, you're going to complete it in class, keep it in your binder. These are more exploratory, like you're going to go through them all and really do the exploratory work because by the time you get to the pig, you're going to get graded on it. And essentially the way this pig is set up so that it's set up like what they call a lab practical. So very often, like in college, you'll be given something like that in a lab where they'll say, you're going to do the following and you're going to set this up and show me what you've got. And then you're going to leave and you're going to get graded on everything. And your teacher comes by afterwards, your professor will come by afterwards and go through everything that you've labeled or set aside. Like sometimes it's slides, like for a histology class, it may be slides. And you're going to have to look at the slide and tell what kind of tissue it is. Is that muscle tissue? And if it's muscle tissue, is it cardiac, smooth, or skeletal? And you'll have to identify it and write all that down. And then your teacher comes by later and just grades the whole thing.
Do you have these dissections like just sitting in your house or they call them?
They're up here. They're in the schoolroom. All of our specimens are here. They're all up in the schoolroom, and there's a dissection kit for each group. But they're all up there. So the sea urchins come in a big bucket. The worms actually came in like a jar. It's all different. Some of them come in little bags. When I did cats in college, there was a big refrigerator, and they were literally like sharks in one side and cats in the other, and they're just like in vacuum sealed bags with the fluid around them, and they were just stacked. It was just like this stack of cats. You could go in and pick your
How
cat.
many sharks do you guys
So-
want?
And I picked the little cat because I thought, okay, this little cat isn't very fat. And I think this cat will be easy to dissect because I don't want a fat cat because I don't want to deal with an adipose, right? I don't want to deal with that. One of my friends picked this big giant cat, and the thing, when she got into it, it was completely, how do I describe it? It was missing organs. It was missing part of its digestive tract. Its diaphragm was incomplete. It was really wild. Like she cut it open and she was like, excuse me, Professor Gray, like what's going on? And that cat was an anomaly. It was an anatomical anomaly. It was born missing parts. But the interesting thing is most of these cats, the cats that they get for labs are obviously cats, they're strays, right? They're strays that are put down and then they're sent to a place where they go ahead and do the preservative process on them so that they can have some use rather than just putting down a bunch of strays and putting them in incinerators or whatever they do, which they do a lot of time. So these have some use. So a lot of these cats are stray. Most of them are stray cats, but this cat was a big cat and he was missing organs. And by his teeth, it was an old cat. So it's a cat that our professor was like, this cat probably couldn't really hunt much because his lungs were smaller and its diaphragm was incomplete. And to do that sort of work, it would need to breathe well and it would need to get a lot of oxygen in its muscles to chase things and hunt. So she's like, I don't understand how it could have survived as a stray because that would have been really difficult for it. And it's an old cat. So she thought it was probably a cat that had belonged to someone at least some point in its life, because it would have needed someone to just put food down on the floor for it, and that would have had a nice, easy, lazy life. And that's how it got to this big, fat, giant cat with missing organs. But it was a mystery because nobody came for it. Nobody looked for it, obviously. And it was put down
Nobody
and
was
sent to the
checking the
science.
pages?
Yeah. So odd. We were well puzzled about that cat. And my cat had a stomach full of worms. So roundworms.
That was
That-
exciting.
But mine was very little. My cat was like this big. And it was great because it was young. So it was easy to separate tissues and things. It wasn't full of fat. Mhm.
Now, the pigs, were they like slaughterhouse rejects or something like that? Like,
So,
what's theirs?
I'm unsure of the harvest process for fetal pigs. I really am. I don't know if they specifically raise them for this or if they're a byproduct. I haven't looked that up, but I will tell you there are thousands and thousands and thousands of them that you can get your hands on. You can go to Amazon and buy a fetal pig if you want to do your own
I just
dissection.
don't
Home, they're
wanna,
there.
like, it feels weird.
It-
A pig that didn't have anything wrong with.
Yep. And I don't know if it's, like I said, a byproduct of the slaughter process, or I'm unsure. I really am unsure for that. Things like the squid and stuff, they're raising them for it. They're raising them for this purpose. And the perch, they're raising, they're not wild or anything. But I'm unsure about the pigs. Kind of afraid to look. I should look to answer your question. But well, hang on, let me look. How are fetal pigs sourced? Let's see.
Right after I said.
Let's just
I'm
go
not
there. Let's just go there. Let's see, because that's important. I gotta move this though.
What? I wanna know.
Well, she wants to know. She can know. Keeping that up. Okay, so
I
fetal
saw the plants and the
pigs
leaves, I kept thinking of that
sourced.
SNL sketch where the guy put eyes on his leaves.
Okay. Well, Wikipedia says,
Oh,
let's see what Wikipedia
That's
says.
great.
So Wikipedia says fetal pigs are unborn piglets of sows that were killed by the meatpacking industry. These pigs are not bred and killed for this purpose, but are extracted from the deceased cow's uterus. Fetal pigs not used in classroom dissection are often used in fertilizer or simply discarded.
Okay, cool, fertilizer, that's cool.
Fertilizer.
Oh my goodness. Okay. So yes, there's sows
Why
that
would
were
you
going
look
to
at
slaughter.
Why would you do that?
I don't know why they allow the sows to get pregnant, though. That seems odd to me that you would put them in a situation.
It seems like a situation where
Yeah.
it's like, they're not meant for this, like. I think a lot of the stuff I eat has fertilizer, so.
And-
I'm never
mhm.
gonna trust fertilizer again. No.
Pigs with mixed sex.
Like this fertilizer doesn't contain.
And then
fertilizer.
here, why are they good dissection specimens? Okay, so they're good because they're mammal-like humans. Nearly all the structures are the same, similar anatomy to humans. Interesting differences. Let's see. They're not bred for the purpose of dissection. They're a byproduct of the pork food industry. Not killed for the purpose of dissection. Those that are not used, there you go, fertilizer. That's disgusting.
Fertilizer.
Most people do not think of pigs as pets. So fetal pigs are relatively inexpensive. They are even at a large size where structures are well developed. So and then all these advantages of dissection, and I think that there are a lot of advantages to actually doing the dissection yourself versus doing an alternate. If you need to do an alternate, that's fine. But actually seeing the inner workings of something. And for me, like some people look at it, oh, it's so cruel. Like the last time we did frogs, I posted pictures on Facebook and I had like a friend that attacked me. That is so disgusting and unnecessary. And I said, I think it's beautiful. You might look at those frog guts and think it's disgusting. I honestly think it's beautiful. The fact that those structures are so perfect and that they all derive from one little fertilized egg and one little single cell zygote, and it becomes this diverse bunch of systems that coordinate with one another perfectly so that a living organism can grow and reproduce and have all of these varied functions is absolutely amazing to me. And to me, it's a beautiful thing. It's a beautiful thing. So I don't look at it as gross. I look at it as just stunning, like perfect. So, but that's me and I'm weird. So I will admit that not everybody looks at it that way, but I think it's pretty cool. Any questions about anything coming up? We're all good. You got about 10 minutes till Spanish for those of you that do Spanish. If you If you're not sure whether or not your seeds may have tumbled to the bottom of your CD case, you might want to check before you go. And if that's the case, you may just want to add like one more layer of paper towel or something so that they're really squashed against the plastic lid. Then she'd be good. All right, later, Sid. Yes.
I'm I can